Lions Rock Productions.
This is Jay Moore.
This is Greg Cruz.
This is Dan.
This
Stone.
is Dexter from The
This
Offspring.
is Nathan
This
East.
is Sebastian Younger.
This is David Lab.
This is Stuart Copeland.
This is Mick
I'm
Gillette.
Christopher
This is
Knopf.
Dr. Bob Greenberg.
I'm Laird Hamilton.
This is Jonathan Sebat, director of the Beyster Center for Psychiatric Genomics at UC San Diego, and you're listening to The Break It Down Show.
And now, The Break It Down Show with John Leon Guerrero and Pete A. Turner.
Yes, and now, man, am I in over my head now. Holy cow. I'm riding solo. I'm down here at UCSD. Yeah, we're at the Beyster Center learning about the impact of genes on autism in this case. So let me start us off by saying, first off, you can capture my man, Jonathan, over at Sebat Lab, at Sebat Lab, Sebatlab on Twitter. He'll talk to you there. You can grab me at P.A. Turner. I will get you over to Jonathan if you have questions. And of course, you can go to the website. That stuff will be in the show notes. So, but it's the Beyster Center. So look up Beyster Institute, and you'll get right into where you need to be. Okay, let's. Here's the thing. We got to start off with this. And you made this point when we talk about autism and all the things in that world. We have to talk a little bit about vaccinations because look, I deal in a world of perceptions, and if your reality is vaccinations are going to harm my baby, then we need to talk about that first off. So give us an idea of the vaccination link to anything in the autistic world.
So the original proposed link of vaccines to autism came from a paper by a gastroenterologist named Andrew Wakefield that was published in the early 90s. And that paper actually didn't find a link between autism and vaccines. It was an anecdotal report of a series of cases where the parents reported symptoms of autism in their kids, and parents also reported, oh, by the way, we started to notice these symptoms around the time that they'd had their vaccines. Okay. And that was it. It was just purely a self-referred group of families that had their own theory, right, that their autism may have been caused by the vaccines. That is there's no scientific link there. That was the only link. And then Andrew Wakefield did a descriptive study of these children, just describing their symptoms and characterizing them a bunch of different ways, and some ways that were somewhat unethical, and some data that was fabricated. But in the end, the scientific misconduct was kind of not even relevant because the original report didn't have any scientifically credible links between autism and vaccines. But what really happened at the time was that the Lancet paper came out. It was in a prominent medical journal, and he somehow ended up on 2020. Yeah. And when he got on 2020, he went rogue and started basically going totally off script and speaking as if they really had found a credible link between autism and vaccines. And, of course, that resonated. We hear that word a lot lately. Right. Resonated. So there were a lot of people out in the community that were very primed to find a culprit. Yeah. And that makes sense. And when Wakefield gave the public this scapegoat, it went wild. I mean, this is basically pre-internet. Right. But basically, it went as viral could be in the early 90s. And it
being on 2020 is pretty viral back then.
2020 was basically it, right? Yeah. So it became a viral meme, basically, that sunk into American consciousness and global consciousness because people were really terrified, and they were one of the reasons why they were primed to believe in a vaccine as a cause was because it's a universal experience among parents whose children get developed with autism. That by definition, autism is diagnosed by age 3. Okay, and the diagnostic clues of autism are failure to meet normal milestones in your development. And guess what? Those specific milestones that pediatricians look for are happening at exactly the same time that pediatricians are administering the standard vaccination schedule.
That's when you go see the
right.
doctor.
So when you go to the doctor and get your vaccine and get your checkup, and the doctor notices that the kid is not meeting these milestones, then suddenly the parent is primed to think, oh my gosh, at exactly the same time that I'm going to the doctor, giving my infant child this shot, which is very traumatic for the child and for the parent.
Yeah, you're hurting
then
my
they
baby.
come home. They come home after having this traumatic experience, and then and on top of it all, the doctor's telling them that now we're not meeting milestones. And so you can imagine how how in American consciousness there was this, primal sense that something is happening, something's going wrong, and we have to find out what it is. And when this Doc Andrew Wakefield basically proposed vaccines, it turned into a massive health scare.
I mean, it makes sense, right? Because you've got your kid isn't okay, and you're trying to put it together, and you're looking for an answer that largely, at least at that time, especially, wasn't available. Right. And so when someone offers something, and we've proven this over and over again, humans are great at setting patterns, but we're shitty at going, oh, that was wrong. That pattern was wrong. Totally. I mean, I can do examples of all day long of the times when we've gotten patterns wrong, and then our own ability to see that problem. It's really tough. It makes sense. But why does Wakefield make that leap? He doesn't have something that's repeatable. It's not replicatable. I mean, these are the tenets of scientific methods. So why is he doing that?
It's unclear why he has basically gone full on into, promoting this fear-mongering and health scare that he continues to do.
Wow.
In a way, I can only speculate as to his motivations. I mean, really, at this point, he's been completely discredited as a scientist and a physician. His medical license has been taken away. So I guess he doesn't have a lot of other options.
I guess that's how he makes his money. He's doubling down.
Right.
Well, I mean, but there's things like that are out there that we struggle with despite the science. The human impact on, and I hate to call it climate change because that doesn't define anything. Like the climate always changes. It's always ebbed and flowed and up and down and everything. So, but there is science that says, hey, we are doing things that harm our environment. Okay, but we struggle to accept that. It's hard to prove the link between GMOs and cancer. You look at the science is like, we're just not finding it. So, and then maybe there's science going the other way, but the predominant stuff, last time we had a show about it, they were like, it's like 90+%. Nobody's finding anything there. And we hate that. And then now we have this thing with vaccinations. Like, well, of course, you're sticking a needle in my arm. And as a guy that's worked in Iraq and Afghanistan where the scientific knowledge is even lower, they absolutely think that we're inoculating them or giving them something that's terrible. Or even like a fingerprint scanner, beep, like a little thing like that that we use all day. I have one right here on my laptop. That is meant to harm you, and so really we're in the same world as that. What the heck? I mean, you guys have this big, huge building here. It's not all dedicated to that, but all this work is being done. I'm sure you're not the only place working on this problem, but no one reads academic papers anyhow. Right. It's like only the crazy, sensational, wrong ones.
Well, basically, in order for the true understanding of what does cause autism,
right,
and how do those causes actually produce the actual symptoms that I'm seeing in my child, in order for people to actually be able to relate to that, we really need to get the basic research, the basic science that's been done over the last 15 years directly into the clinic.
Yeah.
And it's absolutely, from a technological standpoint, we're already there.
Okay.
At least in terms of being able to assign a real causal diagnosis. So identify a cause and assign a diagnosis from that cause. We're already at a point where we can do that in 25 to 30% of cases. Being able to get that technology directly into the clinic such that a significant proportion of parents are actually being able to benefit from this information. That's really what it's gonna take to actually shift people's perceptions and to get the science into America's consciousness.
Okay, so now on the ground, like the reality is you've got a kid that requires direct parenting constantly, because of where they are in the autistic world, and that impacts a lot of lives. Those folks are looking for the day-to-day, like, how do I get my kid to calm down in situation 9, it's on the parent. You're the adult. You're the one that has all the communication ability, and they have one channel, and you're trying to learn. And that channel has never been studied or mastered by anybody. It's been invented right here.
Right.
So what do we tell those folks that are really in it every day?
Well, I deal with this all the time because we recruit families from the community. We sequence their genomes. And in some cases, we're actually able to give a genetic diagnosis to the family. But I'm still stuck when we have the family come back into the clinic and we communicate the results and say, okay, your child has chromosome 16 P11.2 microdeletion syndrome.
Right.
That's great. It tells them, first of all, it wasn't their fault.
Right. Yes.
Number one, there's nothing that the doctor did. There's nothing that the parents did.
Right.
That was the underlying cause of this cognitive impairment. The cognitive impairment came from a genetic glitch.
Right.
That occurred and influenced brain development and influenced behavior in a way. So, what do you tell that family other than here's how it happened?
Right.
And obviously, it's nobody's fault.
Right. It's not because you're drinking milk in utero or that kind of thing. Yeah.
The way human genes work and the way human development works is it's heavily genetic, and it's not something you can control.
Right.
It's a basic process of human development that was basically owed to some genetic glitch in a child's DNA. Yeah. So how do you actually tell that family other than giving them some peace of mind? Which the genetic results do give.
Right.
How do you actually do any real benefit to them? How do you actually make it easier to parent their child? How do you improve that child's performance in school?
Sure.
And how do you make their social interactions more easier?
Yeah.
And that's
you're being
a
very
harder
careful
problem.
to how you pick your words too. Like, you're not saying make the kid better. You're like, how do you improve these aspects that are like, because you have what you have. You have this shaped bucket. Only this much water can go in. You still have to carry water every day though. So how do we work now at this mission? I get that part. Let me ask you this then. So when you look at the genome and you start to break things down, it used to just be autism. And then it was this condition, and then it was a spectrum. But you're saying it's even more individualized still, where you have glitch 75 with a Charlie identifier, right, which we don't see very often. But are we still discovering new forms of these glitches?
Absolutely.
Okay.
We're really just scratching the tip of the iceberg at this point.
Right.
We've talked a lot about personalized medicine.
Right. Yeah.
And from a clinician standpoint, personalized medicine is, really try to understand the genes are part of it. Try to understand what their genetic predispositions are, but also understand more broadly their medical condition. Try to integrate all that information and to come up with a personalized treatment plan that's going to work ideally for you. From a geneticist standpoint, that whole concept is basically the same goal, but we're going it another step further in that we're personalizing it to the level of the individual.
Okay.
Because you really can, in a subset of autism, identify a mutation that is unique to you.
Right.
And that is not shared by very many other people in this country.
Yeah.
So there are some diseases that we've diagnosed from genome sequencing. Basically, you would estimate that there might only be 12 kids in the country with that mutation.
Yeah, out of 330 or so million people. Okay. and when you have that many numbers collide, you're going to have 330 million outcomes, and especially with the way genes go together, it's actually an exponential number from that where you have all these things. All right, so let me back up real quick. And is there any kind of link between vaccines? Like, there's a kid in my hometown where the belief is that he got a vaccination and then he was given a malady. It's hard to tell that family and know that's not the case. Are we looking at something that's not autism maybe? Or how do we reckon with that?
So you're asking when there really is a case where a family member, where a child is given a vaccine and then immediately sees deterioration.
Sure, yeah.
So how to deal with those types of cases?
Is that still autism, or is that
so? Those are not cases of vaccine injury.
okay?
Right. Those are generally cases of adverse responses to vaccines are documented all the time. Many of them are not actually directly caused by vaccines. They're just documented, right?
Okay.
You have to record when there's a possible adverse outcome, and there's a database for recording these things. But in fact, most of the things that get recorded as adverse events are actually not directly caused by a vaccine.
So even still, okay.
Now, there are, you can't deny that there are the potential to aggravate. If you get some shots and you get a fever, you could temporarily aggravate a condition.
Sure.
And that may occur in some cases.
Right.
There's a genetic disorder called Dravet syndrome, which is caused by a defect in a sodium channel called SCN1A, and it causes epilepsy. And so when kids are given shots, they do have a tendency to run a temperature, and that could increase the chances that they do have a seizure. That's in a very rare specific disorder, but it's the underlying cause in that case is the gene.
Okay, right.
And the epilepsy that they're predisposed to by the gene. And so the shot could precipitate a seizure, but that's not the cause.
Right, it's not the cause.
And the kid's syndrome would not have, failed to manifest because of if they'd avoided the shots. And it's not how it works, right? The genetic disorder is the cause of the syndrome,
right?
And so Dravet syndrome kids actually do put themselves on a different vaccine schedule and or in some cases go unvaccinated just to avoid the potential risks, and they rely on herd immunity in our population,
right? The other
so that we don't have whooping cough and we don't have measles floating around in our population,
right?
So people who there's the very rare examples of people who are at risk,
right,
and might actually suffer an illness because of a vaccine. Those are the ones that need to be identified, and they should maybe avoid vaccines or have a different vaccine
and
schedule.
Rely on the rest of
but
us.
everybody else needs to be vaccinated because we can't have these preventable
Oh
diseases floating around in this building.
Yeah.
Or we'd all, all of our kids would
for
suffer.
the large part, cholera doesn't kill anybody anymore, except for in really shitty places, where you're like, there's just nothing we can do here.
Exactly.
Right.
So we've gotten off topic of autism per se,
right.
but I think the discussion of people's perception of their kids' condition and the medical community, I think it really does have to evolve. We've reached a point now where it's now very clear that genes play a big role. You may not be able to give a genetic diagnosis to everybody yet because, like I said, we don't have the full picture yet.
Right.
We still have
on the iceberg. We're just not in the middle of it yet.
Science is a long game.
Yeah. Yeah.
You have to step by step put together the pieces of the puzzle,
right.
and the genes are the cornerstones of understanding what is the array of pathology, what are the array of different genes and biological processes that can go awry in our population that has this diagnosis, and then start to construct from those little cornerstones, try to construct a more comprehensive view of how the gene interacts with development of the brain and how that can affect a child's own perception of the world and how children interact with you as a parent and interact with their friends and how that affects their learning and how that affects their school experience. So there's a lot of basic science, and it really comes down to basic science. You really have to do this. You need to have a fundamental understanding of human brain development and how genes, in order to use that information to come up with a new treatment for autism or for particular syndromes within the autism spectrum. So we really need to invest into these basic research, which brings us back to your original point about the family who's struggling and needs answers now and needs a strategy now on how to cope.
Right.
And that's where the basic scientists like myself are really struggling because we realize that there's a lot of work to do to really understand, even just for a segment of the autism population that we can actually define genetically.
Yeah.
There's still a lot of work to do to understand how the development in the brain is altered and how we can potentially counteract that, either through behavioral training or through medications. And there's a variety of strategies that we have to come up with.
On the parent side too.
It's a long game. It's a long process to get from where we are now to that point.
I want to, and I don't want to stay on the vaccination side forever, but I feel like the audience is begging me to ask this question. What about a bad batch of vaccinations? I mean, that can't still, I understand that can't give you autism, but it can give that reaction for your body, whether it's allergic or it's just some kind of like, there's a bad batch and that happens. Is there any credible way to look at that and go, yeah, there may be something there with that? Or is a bad flu shot, that can't cause, it's not causing autism, but can it cause something like that where your brain is scrambled in a way that you lose your ability to operate?
Well, I can't really comment on the quality control that the vaccine that the pharmaceutical companies use in approving batches for the testing and approving batches. I can only comment on the research, which is that if there was a bad batch that was tied to a severe adverse outcome in a group, it wouldn't be hard to actually make that connection,
right?
Because you can connect.
There's a batch number,
right? Exactly. That's standard epidemiology. And that's the first thing that epidemiologists look for when they're trying to find an environmental cause of something.
Okay.
Is they're trying to trace this outbreak,
right,
to some source,
right?
And in the case of vaccines, you can trace the batch of vaccine. And if there was a bad batch that caused an outbreak of something, whatever,
right,
there are ways of actually tracing that. And so, but what we're seeing in the population is we're not seeing that. We're not seeing.
Okay, that's the main thing.
We're not seeing outbreaks of autism that are tied to things like this, right? So when you look at autism in the population, it's very broadly distributed. It doesn't vary very much geographically.
Yeah.
It doesn't vary very much by country.
Got you.
And when you do see variation between, so like I believe Arkansas has a below average rate of autism diagnoses, and California has an above average rate of autism diagnoses. And you can start to try to drill down and understand what explains those differences. And the science right now does seem to suggest that you can't avoid the influence of access to services, like that. Access to services, and so your ability to recognize autism and your ability to diagnose it requires access to services. Yeah. And if there's differential access in one community versus another, then you can get these discrepancies
for sure.
And so you're trying to evaluate causes of autism on this background, where there are socioeconomic forces that are really kind of driving things, and then you want to try to detect. It's kind of like looking at an astrophysics experiment and trying to detect some signal of birth of a galaxy or a black hole in the background noise of all this other signal that's coming out at you.
Dr. Keating reference.
You have to you have to be able to explain away the noise.
Well, and at some point,
in order to find the true environmental causes, and so that's just where we're struggling with that now.
You get to the point. So in my end of things, I deal in culture at a pretty high level. And the way I define it, and like defining culture, it's a scientific thing. We got to define our terms. You go ask 100 PhDs to define culture, and you're going to have 500 answers. Like they're not going to agree with themselves. So the definition I use, and I think it's pretty practical, and it's of course not perfect, but anything not explained by DNA. Like that problem is a social, cultural problem. Like someone's got a cousin who lives in Palo Alto and she's a maid. And she's like, I work for the guy that fixes autism. Come out from Arkansas. And then mom gets up and go. Like how do you account for that? Well, you got to use big numbers. So, okay. We've seen the number of cases go from 1 in X to now 1 in 60-ish. Depending on who you're talking to. Well, let me ask you this too: is it 100% for sure every case of autism is genetics, or is there like some kind of weird factor way at the end of the wiggly end of the curve where weird stuff happens?
So it's possible to measure in a population how much of the risk is coming from genes, and the ways that this has been done going back to the 80s was to look at twins. And you ask, if you take a pair of identical twins, which are kids who came from the same fertilized egg,
right,
and it divided into two individuals,
yes,
and now they both have exactly the same genome,
right,
and so you compare when one of those kids has autism, what is the chance that his or her identical twin also meets criteria for autism? That's a measure of how much genes play a role, and then you compare that to the amount of what we call recurrence risk in regular siblings, or fraternal twins, which are basically siblings who were born on the same day.
Same day, yeah, right.
And then look at the general population recurrence risk. So general population is like 1% recurrence risk. Siblings is about increased by about a factor of 10. So it's about a 10% recurrence risk. If a child has autism and they have a sibling, it's almost about 10% chance instead of 1% in the general population.
Right.
So being 50% genetically identical to somebody increases your risk by a factor of 10.
Yeah.
Now, when you go to the identical twins who are 100% genetically identical, that risk goes up to a 70% recurrence risk.
Oh, wow. Okay.
It's very large. And so you go from 10%, you go from 1% to 10% to 70% as you move toward being half genetically identical to being full genetically identical. And so the estimates that you get in terms of the proportion is roughly 60 to 80% of autism is explained by genes. Now at the level of the individual, see, that's where you start to see the broad diversity of autism and the diversity of causes.
Right.
Because in some kids, the underlying cause that you can say that this person came into the clinic, almost 100% of the reason why this person is sitting in front of a neurologist today has to do with a single mutation that we found in their DNA. And that's a case where you would say, overwhelming majority of the risk in that individual is genetic.
Right.
And then you have other kids where you come up empty-handed with your genetic testing and you don't have an answer. It doesn't mean that there isn't any genetics there. It just means you don't understand it.
Right.
In that individual, and there may be it may be less genetic in some individuals than in others. So one of the things that we're starting to see, so these mutations that I keep referring to, I should probably describe a little bit more about what they are.
Okay, let's do it.
So the way you find the easiest way to find a set of causal mutations in a group of kids with autism is to look at, to sequence their entire family, to sequence mom and sequence dad and sequence the child. And then you compare the genomes of the child to the genomes of mom and dad.
Okay.
And where this genetic signal is really concentrated is in the small number of mutations that are actually not inherited from mom and dad, but actually occur as new mutations in the child. And we all have a few new mutations.
Sure.
That's how evolution happens.
Right. Yeah.
Every generation in your family, there are new mutations that occur that weren't there in the parents, and that's very normal. We all have 60 to 80 of these in our genome.
Out of how many? What's 60 to 80?
Out of a million variants across the entire genome.
Right. Okay.
In your entire genome, there's like 60 of them.
Right.
That are new mutations that you didn't inherit from your parents. And if you focus in on those, it's actually very easy to see. The ones that disrupt important genes occur much more frequently in autism than they do in the population of typically developing kids. So you can see a huge enrichment of mutations in these developmentally important genes that we know have a function of brain development. They're expressed in the brain, and they also are very resistant to mutation. They're not tolerant of mutation. You don't find disruptions of these genes in the general population very frequently because they're very deleterious. Right? There's a very strong natural selection against these mutations.
Sure.
And so these are what you call clean genes. The genes are very clean in the population. You don't see a lot of variation in them. And then when you see a de novo mutation that disrupts that gene. There's a huge difference in the frequency of those in autism compared to the typically developing siblings. And we also see enough mutations piling up in individual genes that we now have a couple dozen unequivocal, these are disease genes, and we can see that these mutations are disrupting them, and those are actual genetic, those are genetic contributors.
Yeah.
So that's the kind of stuff that we can test for now.
Okay.
And I forgot what the original question was.
So we were getting into is 100% reliability on genes. That's
So in the type of mutations that I'm talking about, that's maybe in 30% of autism. We can say that this explains a large proportion of the risk in an individual.
Yeah.
And when you look at that subset of kids, they are more impaired than the broader autism population. So if you took autism and you divided it in half into kids with below average IQ and kids with above average IQ, these mutations would be concentrated in the below average group. So these are kids who are have a heavy genetic load, and those types of mutations that I'm talking about have significant effects on brain development. And they tend to form a group within the autism spectrum that tends to be lower functioning. Now, when you look at the kids who are higher functioning that have above average IQs, you see less of this genetic signal, which comes back to the point of, is it less genetic?
This episode of the Break It Down Show is brought to you by Lions Rock Productions.
That's us.
We publish, evaluate, and develop podcasts just like this one, consult others to build their own, and create associated content and content marketing strategies.
So if you're launching or expanding your social media presence, your business or your personal brand, or if you just want to take your media presence to the next level,
reach out to us on Twitter
at Pete A. Turner
or at JohnLG69
at the Break It Down Show.
There's 1,000 ways to get a hold of us.
Now enjoy the show.
Right.
Very well, possibly, but it's also less autistic.
Yeah.
At the same time. So these are less severely these are less severely affected people who have a more complex presentation and also have a more complex etiology, right? So there's not a simple monogenic explanation for that kid, because that kid has a very different type of presentation.
Yeah. Well, I mean,
and those are also the kids who are going to graduate from high school with honors and go on to college and get a college degree. So this is a different class of autism who has their own struggle.
Right.
But obviously don't have the same level of impairment as kids on the opposite end of the spectrum.
So, okay, let me I'm holding my face together because I want to make sure I ask this question in the right way, and I'm at the edge of my ability to do that. So, okay, we understand that there's, and I'm going to use the best words that I have, there's the quote-unquote idiot savant who's like, this guy is autistic, but he can do whatever task that nobody else can do. Just because you have autism doesn't mean you're that. That's part of this whole spectrum thing. And I would submit, anytime I hear the word spectrum, I think it's really more of a sphere. Where maybe socially you're normal. You may appear normal, act normal, but in a given situation, you are no longer a normal kid. Now you're somewhere on the spectrum. So really, you're somewhere in this spherical realm, and I'm assuming that you would say that there's going to be bubbles within the sphere, and like these things are all captured within this genome thing. And then you work in the 30%, like a third of the autism realm is an area where you can play in, and the predominant answer is I can find a genome type that corresponds to what you're doing, and then the things outside of that we don't know. It could be something we haven't discovered. It could be genome still, we just don't understand how to look at it and see it, and then it could be something even beyond that. So you haven't said no to any of those things, so I feel real smart.
That's a very clear and accurate description.
Okay.
We have ways of now starting to tease apart the complexity.
Yeah.
And to understand these higher functioning cases, because as you get more statistical power through having more and more data, then you can start to pick up the more subtle signals, and so you can start to understand the other things that explain the other 70%.
Yeah.
I don't know that we're ever going to get to 100%.
Sure.
Autism is not 100% genetic. It's 60 to 80%. And so as you start to move away from these things that look like what we call monogenic forms into the more complex, you start to have to be able to understand the interplay between one gene and another.
I got you,
the interplay between multiple genes, and the interplay between genes and environment. That's a long game. That's a process where we have to go incrementally in understanding the different modes of inheritance and the mechanisms that underlie those. And that's where we're going in the future, to try to make genetic testing more informative, to improve the diagnostic yield, and most importantly, to be able to offer something to these families in terms of what you can do.
Yeah.
What you can do to help improve, the cognitive performance in this kid.
Oh my gosh.
We're moving all in all of those directions.
Yeah.
But we need to take it one step at a time.
And there's so many great questions, so I'm just going to do my best. And I'm really trying to answer the questions for the people in the audience, because here's my next question with that regard is, you play in 30% of the autism realm, so you have peers that overlap or are completely different, and are they finding similar numbers? We don't got to get specific, but a similar vein where, like, yeah, we're learning more and more about genes. It's more and more about the genes. Is it fair to say that?
Well, so you don't really divide the population into, this group we study and the other group. We actually sequence everybody.
The population.
Right.
Okay.
So we sequence everybody.
Okay.
And we're only successful in that 30%. And we're unsuccessful in the other 70% in being able to actually identify a clear cause. But it's not as if we're studying different
But that
groups.
number's
We're all
waning.
studying the same population.
Got you. Okay.
Other people that are asking different scientific questions may be able to gain some insight into that other 70%.
I got you.
I think that's where you're going with this, right?
Yeah, well, I didn't know, like, if there was like a type A, type B, type C kind of field, and you guys are playing in type A, and then there's some A, B'ers or whatever. But if you're saying you guys all deal
We're all
with
studying
the
the same populations.
Right, I got you.
But with different strategies.
Right, okay.
And that you're absolutely right that someone who's studying maternal viral exposures, maternal infections during pregnancy,
Right.
May be able to capture something else
Right.
that we can't capture with genes. And so there are groups who are starting to look at that epidemiological data, and obviously once successful, so there is some evidence that suggests that the small amount of there's a small increased risk if mom had a viral infection while she was pregnant.
Right. Okay.
That there's a subtle increase in risk to her child for developmental problems, including autism. So the problem is that those epidemiological studies have nowhere near reached the same level of power that our genetic studies have,
Yeah.
and so they need to catch up.
Yeah.
So once you can start to do this at the level of individuals. So let's imagine we're all studying a population of 1 million people.
Okay.
And there's a group who's studying the maternal viral infection. There's another group who's studying the genome, genetics, and there are other groups who are also looking at nurture and nature in the context of, socioeconomic factors, parents' IQ, parenting strategies, single parents versus nuclear families, and all these other socioeconomic social factors. When you can eventually start to integrate those, you can start to look at how they interact or how they don't interact. Maybe is there such a thing as an environmental autism? And can you take an exposure, define a group of kids who have had that exposure,
a common exposure,
and
right.
now it's a very simple scientific question now to ask: is the genetics reduced in that group?
Got you. Yeah.
In a way that a matched, is there less of a genetic load
Right.
in that group than there is in the rest of autism? That's a very testable scientific question.
Yeah, for sure.
And we need to get there. So the epidemiology has to get to the point where they can actually label a group of kids within our 1 million kid cohort,
Right.
and say these are the ones that have had the exposure, and it's very straightforward for us to then test whether that you would predict that if that's an environmental cause, then the genetics would not be any different in those kids than in the general population, right?
And again,
we can test that.
Yeah, and again, like that, you could take that back to, say, the bad vaccination batch thing. You could look and see, like, is there a correlation there? And, like two doesn't make a correlation, and it takes more than that. So okay, I get that stuff. And man, that was like such a great picture. And I know there's a whole bunch of other questions in there, but I want to talk more about genes in general, because this stuff has become. There's your field, but I mean, how many gene-based fields are there now? Like, we just caught a murderer with 23andMe.
Oh, we have a few of them.
Yeah. We won't release or if we know that to be true, why aren't we leaning heavily on DNA? Like, there's the guy that's on death row at San Quentin, and there's viable concern as to, like, did this guy actually do it? There's the case, he was up in Corona. Do you know about this case at all? Anyhow, the DNA, the evidence, they found a drop of blood later. And they're like, yeah, let's test this. Yep, it comes back with his type. But every indication in that blood, now that we know more, is that blood was in a vial before because of how they housed the vial. They're like, that actually didn't come from, because you don't have those chemicals.
Contamination.
Yes. And then they go back to his vial, and there's 2 types of blood in the vial, not just his.
Oh.
Yeah, exactly, right? So the first thing
that would invalidate the test.
Yeah. And so we won't take DNA to figure out, like, okay, no kidding. There's a real problem there when there's 2 types of blood in the same vial, you know? Mm-hmm. Anyhow. So the whole thing I'm getting after here with the gene thing is and we're learning them in parenting. The genetic makeup of the kid has a massive impact over the success of that kid's life and his or her behavior. So yes, you have to provide for your kid and, you know, do things, but really your impact on their behavior and those things is pretty limited when you look at what the genes account for. There's not a whole lot left. Mm-hmm. You know, it seems like. What are your thoughts on that in terms of other areas where genes are really kind of not predestination of activity, but you're predisposed to things because of your puzzle makeup?
Well, just on a more general, I mean, as a geneticist,
yeah.
I think I'm very generally interested in how much of my own disabilities,
Yes.
why is it that when I sit down to read a scientific paper, the first 15 minutes I drool on top of the paper and fall asleep, and then wake up after 20 minutes and then I get into the zone. And now I read 5 papers in one sitting and get, sink my teeth in.
Yes.
And whereas, so it's like, it's a huge amount of work to get over that threshold and then to get into the zone. Whereas other people can sit down and boom, 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 hammer it out and take care of business, right? So, like, why is it that I have my own disabilities and cognitive impairments, and how much of that is actually related to my genes? I would very much like to be able to. I've sequenced my own genome a couple of times,
right?
And again, I don't know the answer to that question.
But it came up that you're an asshole, right?
The asshole gene has not been discovered yet. But
it seems to be all of them.
Right.
Okay.
But there certainly is a link between success in gene discovery and assholiness.
It's one of my favorite jokes. Like, I took a character test or like a personality test, and goddamn, it said you were an asshole. Like, that's such a funny thing. So, okay, and that kind of goes back to the bigger question: is if we are talking about an autism sphere, is that thing going to get bigger? And, like, let's say that's a form of autism that you have. Like, I get to the abstract and I have to drool, and then I have to calm down, and then I can get back in because I have autism type code number or whatever. Mm-hmm. Is that really what we're talking about? Is as we get to understand these mutations, it's like everybody has mutations. You know, some guys got feathers. Some guy can remember every name. Like, you ever see that Stan Lee show where they talk about like real superheroes among us, and the guy's like, I can remember everybody's name on this trail as they walk by. I just need to hear it one time. That's not a skill. That's genes. You know, like that gives him some kind of his memory tunnel is huge in his brain. But is that what we're talking about? Is we're going to get past 1 in 60 and just call it? I mean, that's like, I don't know, 1% of the population. So are we talking like a ubiquitous thing? Like everybody has some kind of autistic mutation. It happens in them. And for a lot of us, social norms and performance norms are there. Apart from like my inability to type and read my own errors, you know, I think that's absolutely related to getting blown up. But what do you think?
Well, I think that so the use of genetics in medicine can definitely go beyond simply giving a diagnosis to the most severe kids with specific syndromes. I think it can go definitely beyond that,
right? And
more into the realm of have your genome sequenced at birth or before birth, and as you go through development and you go through your normal pediatric checkups, make use of the information any way that you can in understanding the child's risk of diabetes, understanding, potential interactions with drugs. Like maybe this kid could metabolize Ritalin faster than another kid. and maybe that's not the ideal drug for use in this particular kid. And it could be used beyond neuroscience. It can go well into many different aspects of medical care.
It can go into party science. You're like, oh, this guy's got tequilanase, the tequila dissolving enzyme, and he's going to be our guy.
That's why this guy can do 7 tequila shots at Senor Frogs, and I can only do 3.
Right. So we're going to put him in the competition for the shots because we, Delta Chi, do or die, or whatever.
Absolutely, absolutely. No. There's a very clear example of how certain individuals in Asian populations are actually virtually immune to alcoholism because they do not metabolize acetaldehyde dehydrogenase. Acetaldehyde is actually the toxin that's produced from alcohol that gives you hangovers. Yeah. And so these people have such severe reactions to alcohol that they just can't drink enough to become addicted. Right. And so you think that this is one example that's known of how addiction risk is influenced by your genetics. Yeah. But this is just one example. You think it's going to stop there? That's just a known example. There's going to be a lot more of our own biology, our own personality traits, and our own susceptibilities. Yeah, sure. That are explained by our genetic makeup. That's it's our blueprint. Yeah. The genome is the blueprint for how we develop as a human being, and there's a huge diversity of that out there. And the more we can make use of that information in medicine, I think is a good thing. And again, like you used the example of the forensic investigations that have used genetic databases to identify serial killers. Right. There's a lot of great ways that the genetics can be used as well, and of course, we also have to come up with ways to protect the information. Sure. And use it responsibly. So that's a whole process, but that's no different. And
there's so much learning to do in this area. Like when you think about, for example, like tolerance to pain, right? I mean, that has to have a genetic link to it. And yes, maybe you can build up a tolerance over time, but it's way less about toughness than what we think. It's more about like, I don't feel pain until 30%. Like when you're at a 3, I'm at a 0, and it's not because I'm tougher. It's just because I don't have that capability that you have, and that can be tied to emotions or anything else. So you could, in theory, really individualize medicine and go, like Laird Hamilton, he's been on the show, and he's a badass, right? That guy's incredible. But clearly he's a
legend around here.
Yeah, he should be. I mean, he's incredible. So, like, he doesn't take pain medicines really ever, walks around with a bad hip forever. But, of course he's tough. We don't want to say that, but there's also going to be some kind of genetic predisposition or programming for him to not feel pain like a person who feels extreme pain all the time, Mm-hmm. Yeah, it's interesting. And what do you do? I don't know, like, as we look into the future in terms of genes, we don't want to be so risk adverse we don't do anything, and there's certainly a potential for that. Like now we can't drink milk. Now everybody's gluten intolerant. Now everybody's like, what can I eat safely? Because it always changes. But what are we talking about then? Like how do we improve? Let's say that I get my pre-birth genomic assessment, and it's like, hey, he's pretty smart, he's pretty athletic, he's really messy, and all these things. Do I have the ability to defy my genes, or do I pick the path of these are the things that I am, and these are the areas where I need to focus on either improving or accentuating my strengths?
The genes basically are it's the hand that you're dealt when you're born, but and they have a powerful influence, but it's not as if it's all completely deterministic from that point on, and there's no way that you can modulate how they interact, and there's no way you absolutely can make lifestyle choices, and you can intervene in ways that could potentially overcome some of your challenges that are genetic in origin, or take advantage of some of your strengths. Sure. So that's the name of the game in terms of personalized medicine. It's not as if you're just going to say, oh, you're going to read your blueprint at birth, and you're like, oh, well, that's it. You're going to be a CPA because you're good at math, and you're going to have 1.2 kids, and that's, you're just to wind you up and go, let you go. And it's not like that. Right? That's not really, how life works. Right? So we want to be able to make use of the information to inform our lifestyle choices, to inform what drugs we should take, what drugs we shouldn't take, and to understand what type of autism this is in one child compared to another, and really understand that the impairments can be completely different, and you really need to personalize your care of someone who's actually has serious medical problems in a way that's actually going to correct.
Okay, so here's the
best of your ability.
Here's the big question. So we've had a number of people on that deal in the genetic space, partly from IndieBio on my Popping the Bubble podcast, and also we had Dexter Holland from The Offspring, who he's got a PhD, and he dealt in microbiology. So what I'm wondering is do we get to a point, and I don't know, you'll tell me, maybe it's tomorrow, maybe it's 25 years from now, but maybe it's 100 years from now, but where we can take autistic kid, 27, and you say, listen, we're going to drop these micro or messenger RNA strains in, and they're going to mute out these, it's like it just has the ability to tamp it down, or we amplify the other things that override. Is that coming, do you think?
Gene therapy is definitely coming.
Okay.
Right? So we've had clinical trials of certain disorders that are convenient in the sense that it was the perfect disorder to test out gene therapy on.
Right.
Because, for example, there are certain types of anemia that are caused by recessive conditions where there's an enzyme missing from the hematopoietic cells, the blood cells.
Yeah.
And you have to get some function back.
Okay.
And you could potentially cure the disease by getting some level of function back, because there's zero function.
Right.
The enzyme is gone
in
those kids. And if you can restore a little bit of function, you could potentially cure the disease. And there actually for some diseases of the hematopoietic cells, it's actually working because you can actually insert the gene. You can take out the kid's bone marrow. You can insert the gene into a population of bone, and you can replace the bone marrow, put it back, and actually see improvement. Very recently, my Cold Spring Harbor colleague from my old New York days developed a therapeutic gene therapy approach for spinal muscular atrophy, where this is a particular type of gene that there's different forms of it that are present.
Yeah.
And there's an inactive gene that's just sort of a legacy gene that's stuck in the genome that doesn't actually work. And then there's the regular SMN gene, spinal motor neuron gene, that's actually required for normal development. When the kids have a mutation in the regular gene, it causes spinal muscular atrophy. And you have to just restore some level of function in order to make that happen. And they figured out a way to correct the other legacy gene that other gene in the genome that's just sort of sitting there not functioning. They can actually restore the function of that other gene. And they've had very dramatic results with the early clinical trials of gene therapy and spinal muscular atrophy.
Wow.
So this is not really science fiction anymore. This is current. This is the current state of the art, is that in some diseases you really can correct the genetic defect and restore the function of the gene to the point where you can see dramatic improvements. These kids are living much longer. They used to die in childhood, and now they live. Now they're living much longer.
Right.
So that's the dream. That's what you would hope to be able to do whenever you can identify a genetic glitch that's explaining a lot of the risk in the individual.
Yeah.
How can we restore that function? And it's, spinal muscular atrophy was an example of a horrible disease where kids die very early. Yeah. And they were able to come up with a strategy. Now, that's one very rare disease. And now we're looking at thousands of other very rare diseases where there may be some potential to do that. You just need to develop the technology. So take advantage of similar types of technologies as to what's being used in spinal muscular atrophy. Or now more recently in ALS by my UCSD colleague Don Cleveland. Yeah. Just received the Breakthrough Prize for his work on ALS and the development of a DNA-based therapeutic that can actually restore or basically eliminate the toxic transcript that's produced from the genome and improve ALS potentially, right? It hasn't. They're not there yet, but it's going to keep moving, from SMA, from ALS to a broader range of syndromes where this could potentially work, and we need to start working on it. We've taken a similar strategy in the lab, where we are starting to focus in on those convenient syndromes. Okay, the ones which to us appear to be potentially treatable, the low-hanging fruit of personalized medicine, and now. Let's try to exploit that to try to make a difference in somebody's life. Now, some of my colleagues look at some of the work that we're doing, and they're like, okay, even if you could cure that disease, that's 12 children in the entire country. Right, yeah. You're not really – it's a drop – not even a drop in a bucket. It's like a snowflake in a bucket. Right. It's a really big bucket. Compared to the broader autism population, so what are you really going to accomplish if you can develop a treatment for that? Yeah. And my response is, if you're going to come up with a treatment for, you have to, being able to treat and cure one kid with autism. That's one more. That's one more kid with autism that we could cure than we had yesterday. Yeah. And so you need to have some early success, taking advantage of those early syndromes. Yeah. And so the way we're trying to go about this is to take advantage of this new genome editing technology, which is called CRISPR.
Is that in Microsoft?
No, these are molecular technologies that were developed by Jennifer Doudna and Emmanuel Charpentier at UC Berkeley, and have been further developed by Fang Zhang at the Broad Institute at Harvard. This is a new aspect of our biology that we can now take advantage to actually edit our own DNA. Okay. And so we're doing this in mice. It gives us the ability to put exactly the same mutation that we found in the kid. We can now put that mutation into an animal. Okay. So we're starting with mice, but you could also do this with rats. Yeah. And you can also do this with monkeys. And now you can start to be able to model a specific syndrome. And if you have a treatment in mind, an experimental treatment that you would like to test, you can start developing these personalized models. So we have kids in San Diego who come into our study, get their genome sequenced, and we find that rare genetic glitch. And in a few of these cases, we're like, okay, this is something that we think we can treat. We're going to make a personalized mouse model. We're going to make the Johnny mouse. So Johnny came in, we found Johnny's mutation, and now we're going to make the Johnny mouse. Wow. There may not be another kid in this country with that exact mutation. Yeah. But it's a useful model, for us to understand what are the core neurobiological defects that are associated with this mutation, and then how can we potentially reverse those? Because we cherry picked this gene because we understood a little bit about its biology. So one example would be a neurotransmitter receptor that we're working on that was from a kid named Alex. In our study, and we made the Alex mouse, and now we knew the rationale for doing that was that we knew that we could positively regulate the function of this receptor by using a compounds which we call ampakines. And they have not been FDA approved yet. They have been tested in ADHD and actually have shown to have a benefit in ADHD that's similar to Ritalin. But without the without the stimulant properties of Ritalin. And so that's actually a very promising class of compounds that's been just completely not been tested in the context of autism or to a very limited extent in ADHD. It's a class of compounds that just hasn't been explored. Yeah. And now we found a syndrome that is the perfect target for this class of compounds. And so, okay, let's see if we can actually figure out what are the main behavioral and cognitive impairments that you have when you put this gene into an animal, and then can you reverse those with this new class of experimental drug that has never really been tested before? And that's the strategy that we're taking.
It's incredible. There's a company down here in San Diego also called Stelvio Therapeutics, and they're working on curing cancer. And one of the things they're able to do with their AI is go through, among other things, every existing drug protocol that exists, and then start to crunch the numbers on what would work for John, what would work for Pete, what would work for Chris, whatever, and start to just simply repaint your nucleus to be a healthy and go, because now they can zero in on the cell and go, that's cancer right there. Mm-hmm. And then let's shoot it. Boom. Now it's healthy. And I'm way simplifying it. But all of these things are coming. And so even if it's emerging of your guys' technology where 5 years from now you're like, wait a second, you can do X. Oh, man, I could use that. Mm-hmm. And again, we don't know, but getting back to the whole point of 12 kids in the nation. Yeah, but you can't cure all of autism until you can cure one kid, or reliably cure this group. And it's going to take until we figure out, it's going to take 25, 30, 75 different steps to get closer and closer and closer and make it harder for autism or whatever ailment it is to continue to exist. So, man, I dig what you guys are doing. I appreciate you. Oh, let me tell everybody this. Jonathan Sebat is at Sebat Lab on Twitter. You can also go to the website. It's fascinating what you guys are doing. And I want to ask so many questions about the future. Like, I really want to get a dorsal fin.
That I think that might be possible.
All right, sweet.
Yeah, we can splice in some of the dolphin genome,
right?
And it might actually help you to hold your breath too.
Yeah. Oh, you know who we're gonna have? Well, he's already recorded a show. Is Herbert Nitsch. That guy's a free diver, and you probably know about him, but these guys aren't really notable. But guess how deep the deepest person in the world goes?
I don't know.
800 plus feet.
Wow. How long are they holding their breath?
Oh, like 10 minutes. I think he's like at 12 or something like that. Let me put it this way. He goes all the way down, comes up, is like, I got to take a break for a minute, so I might even take a nap while I'm up. And it's like a break for like a minute. So like it's not even like down and up. He's like, nah, you got to chill out. Otherwise you get all messed up. Mm-hmm. So, yeah, he does not need a dorsal fin. He's already a damn fish. But, yeah, 800 feet is like a quarter of the depth of a submarine. And it's deeper than a lot of World War II submarines, for crying out loud. Anyhow, I appreciate you coming on the show. Anything else you want to throw in here before we wrap it up?
No, I'd just like to really, give a shout out to all of the really creative scientists and members of the autism community that have made this research possible. We don't work in a vacuum anymore.
No.
This is big data. These are big, complex problems that require a lot of different experts to work together, both in terms of the clinic, our colleagues at Rady Children's Hospital, like Stephen Kingsmore, the head of the Genome Institute over there, Joe Gleason, the head of the autism program and neurology over there, working, very closely together with the UCSD academic community and all of the families in San Diego who have really, donated a valuable amount of their time and their actual DNA to make this research happen. So, we hope to be able to take advantage of that and have some more breakthroughs in the near future.
And also thanks to Dr. Brian Keating, who's one on the show and hooked us up. He's Dr. Brian Keating on Twitter. And his stuff is also incredible and has his own episode that's already aired on the Breakdown Show. Yeah, again, thanks. I would love to come back in and check in from time to time. And we don't necessarily got to even talk about autism and genes. We could talk about something else because there's plenty to discuss. But thank you very much for coming on, man. I really appreciate you.
My pleasure. Thanks for having me.